Mustafa Karataş van het Rega Instituut in Leuven werkt aan een methode om virussen op te sporen in afvalwater en lucht. Wetenschappers kunnen zo nieuwe uitbraken zien aankomen nog voor iemand ziek wordt. Mustafa Karataş is genomineerd voor de Eos Pipet 2026. Deze aflevering is in het Engels. Je kan een vertaling lezen op www.eoswetenschap.eu/eospipet 2026. Hello Mustafa.
Hello Kim.
First of all, congratulations with your nomination for the Eos Pipet.
Thank you so much.
You are nominated for using and developing and researching a method that can detect a virus outbreak before anyone even shows symptoms. For me, that's like, in my imagination, that's like a magic ball in which you can see the future of possible threatening viruses. But explain, how does it work?
So obviously it's not magic, but we have to look at different surveillance systems to understand which viruses are circulating where. And traditionally, this is done by using samples that are collected in the hospitals. So one person has to get sick and has to go to hospital and has to get tested. But we can use environmental samples such as wastewater or indoor air, like in our research, and we can look at these samples to actually see what's going on in the whole population or in a targeted setting of different individuals. And this way, we can find viruses that are circulating in the population but that are not making us sick yet. So this is one way to look at it. And of course, there are different methods that we can detect viruses.
So now if we try to detect viruses and infections, we have to have patients and then you take a sample from them, like saliva sample, I think, and then you can determine which virus it is. But in your method, you're just taking a sample of air, a sample of water, and you can find all the viruses that are in it that are possibly threatening.
Yes, exactly. So our method is called viral metagenomics. And I want you to think this as during the COVID, we learned that there is a PCR test, right? This test looks at one virus or a couple of viruses being present in one sample or not. So we get nasopharyngeal swabs, and those swabs were tested for COVID, SARS-CoV-2. What we do is looking at all viruses and their genomic material. And by genomic material, we all carry DNA, and viruses have those too. So we are looking at all genomic material, all DNA in air and wastewater. Obviously, we try to steer our analysis towards viruses. We have to enrich for viruses, which is not easy. But then we have a huge amount of data that we have to analyze and identify viruses in it. So indeed, we can identify all viruses in one sample type. This can be clinical or environmental. And in my case, it's environmental samples.
So you take some air, and then with viral metagenomics, you can detect all DNA that is in the air. So even human DNA, animal DNA, viral DNA. I'm correct?
Yes, that is correct.
And then how do you filter the viral DNA from all that DNA that's in the air?
So first of all, it is correct that viral metagenomics can actually result in other genomic material as well. But we try to enrich for viruses before we do the reading of those genomic sequences. So in the wet lab, we are actually enriching towards viruses. We are filtering our samples, and we are sometimes even using little magnets to pick the virus genomes that we are interested in. This way, we can find viruses, and especially viruses that we are interested in. But even then, we still capture some bacteria or even human genome. But these are very fragmented and not informative. So we focus on viruses because actually they do have very little genomes. So when we capture, we have a lot of information. So everything is there, but we try to steer our analysis and our wet lab procedures towards viruses.
We're here at the Rega Instituut in Leuven. We're in a small meeting room. How many viruses are circulating right now around our heads?
So as you can hear, actually, the ventilation is working. So I'm hoping not so much, but viruses circulating in indoor air, I can say thousands or maybe millions of different viruses. Although most of these do not infect humans, we can imagine these viruses are infecting maybe plants, maybe someone's meal that they had before us here, or maybe some fungus infecting viruses and so on. So I would say thousands of viruses at least. Also viruses infecting bacteria, of course.
How do you take a sample? Wastewater, it sounds obvious for me. You take a liter of water and you do an analysis. How do you do it with air? You catch some air with a plastic bag, or?
So actually, wastewater is not that simple as well. But I will start with indoor air. So indoor air, we collect indoor air using a machine that collects 200 liters per minute. So 200 liters of air per minute. And this machine actually sucks up air very strongly and puts this air through a filter. Then in the end, this filter has many virus particles and bacterial particles and so on, which after this dry filter is being put into a liquid medium. And we do this with our collaborators, Emmanuel Andre and his group, that there are so many different locations they collected samples from. And for wastewater, so indoor air is more like they're just collecting so much air and putting it through a filter. For wastewater, it's complicated because wastewater is very variable. So we have collaborators for wastewater as well, Professor Mark van Ranst and Elke Wollens. They're collecting samples from Leuven wastewater treatment plant, and there is actually a machine to collect these wastewater samples because wastewater is very variable even during the day. There is a machine that is collecting 50 milliliters of sample every 10 minutes. And in the end, after 24 hours, we have a very big composite sample from every 10 minutes. So in the end, we have a huge sample that we have to use for analysis.
Later, we will go on deeper on the wastewater. I want to talk about the air sampling. But one more question about this collecting method in wastewater you were talking about right now. Is there a machine then in the sewers in Leuven that collects these samples? The machine is it down there in the sewers or?
No, it's not in the sewers. It's it's at the wastewater treatment plant.
Okay. Yeah.
So all the wastewater that we are producing goes into the wastewater treatment plant, which there is a machine. And actually all around Belgium and Flanders, there are machines in many wastewater treatment plants. But these samples are only being used for SARS-CoV-2, influenza, and RSV surveillance, so very targeted virus surveillance.
And then for the air sampling, it's like a vacuum cleaner that collects air, and the filter you are analyzing. If this filter, I think you take this filter to the lab, and then what happens then?
So we actually have to get all the viruses or whatever material is in there to a liquid medium to be able to analyze it. And we do that by putting this filter into a medium, which is viral transport medium, nothing very special. Then after that, we have to extract the genetic material. As I said, viruses have DNA or RNA that we have to extract. And after that, we have their DNAs or RNAs. And we have to read these DNAs somehow. And these techniques are called metagenomics or next generation sequencing. So for this, we have a huge genome of one virus, and we actually split these genomes into pieces. After that, we have short pieces of their genomes, and we use a machine for sequencing, we call it here, and that is actually simply reading the genome pieces that we have. So we have to extract it. Obviously, we have to do enrichment for viruses if we want to do it, and then we have to read these little pieces of genomes.
You're especially searching also for viruses that are unknown for science, for everybody. I can imagine if you find DNA from viruses that are known, then it's like matching a DNA passport with virus, and you can name it. But how do you name a DNA code from who you don't know from which virus it is?
Yeah, so obviously sometimes they're not exactly the same, but they are similar. And then we call them variants or taxonomically closely related viruses. So for example, SARS-CoV-2 was not completely novel, or it was a virus that we kind of know. Coronavirus did exist, but different types of coronavirus, we call them species, we might find new species. So they still share some part of their genomes with certain similarity. So this is one option. And the other option is they do not look similar on nucleotide level, so in genome level, but sometimes they produce proteins that are similar to other viruses. Proteins are more conserved, so therefore we can identify novel viruses. And sometimes novel methods actually use just the shape of the protein to determine a virus existence and maybe naming a virus. But in our case, they're usually closely related, and we can tell them they're new variant or not.
How often do you find new variants or new species? Is that almost in every sample, or is that very rare to find a new species?
New species, that differs from one virus to other. So it's hard to say, but new variants or the new strains that were not circulating before, I would say it's quite common. It doesn't have to be very human health related, but we know that there are different strains of viruses constantly being introduced to different countries, different locations, and at different times. So throughout the year, we probably start seeing different strains in the wastewater, although maybe we're not detecting them because it's not perfect and methods can be not as sensitive as we want.
So if you detect new viruses in the samples, how do you know that this new virus or this new variant is possibly threatening, possibly infectious, possibly the start of a new epidemic, pandemic?
So that is very complicated, and it's actually, we cannot say if these viruses are going to cause so many cases in the population. So this is one of the limitations of our studies. Of course, we are looking at viruses that are circulating in the population. We can tell that they are circulating, but it's hard to say that they're going to cause a problem for sure. Obviously, if we know a novel variant, for example, bird flu, that is circulating, that might be of concern. But otherwise, if it's just a completely new variant, it's hard to say that it's going to be a bigger problem than previous ones.
Then you have to keep it in guard and see what happens if people get in touch with those new viruses.
Yes, exactly. We have to connect our surveillance systems to the clinics because by itself, environmental surveillance can alert you, but it will not tell you how many people are actually sick and going to hospitals.
One part of your research was collecting data samples, viral samples, air samples in daycare centers and in hospitals and analyzing those samples. What kind of viruses you detect in daycare centers?
So actually daycare centers are a very diverse group of people. I mean, population is 0 to 3-year-old children, of course, but coming from all around Flanders, potentially their parents are working in a hospital. So we do find a lot of viruses, including human viruses that we expected. Human viruses can be respiratory viruses, so many different types of common cold causing viruses. We call it cold, but there are many different viruses underneath. And we also find, of course, skin-related viruses, which more than we expected. So we are always shedding our skin, right? And we can find these in indoor air. And also, when the windows were open in daycare center, we could detect bee-related viruses or some bird-related viruses that are not concerned to human health. It's just a complete population of viruses in one location and so many different viruses, maybe thousands.
So you also find a lot of viruses in those daycare centers without the children are sick.
Yeah, exactly. And this is something we also heard during the COVID pandemic, right? People were actually not showing any symptoms, but they were still shedding virus, or some people were getting sick without contracting anyone who is sick. So we can have subclinical infections, we can be infected, and we may still shed these viruses. And that's probably what we saw. And we can also shed viruses before we show symptoms or after our symptoms finish.
I'm a father of a 4-year-old, and I remember from the time that he was in daycare center that a lot of parents almost complain about the fact that in those daycare centers, they always get sick and they spread around all viruses. And so the parents also get sick. Do you have the impression that the daycare centers are a breeding place for viruses?
I would like to think it as immune system is developing, because this is a known phenomenon that up to 5 years, children will get sick. And normally they will recover with no big problems. And of course, if you put 20 kids together with different families, and maybe they also have siblings who are going to elementary school, high school, of course, there will be some transmission events. But this should not concern people by itself. We should more think about how can we ventilate all the rooms everywhere and how can we do it better. Other than that, this is, I would call it immune development.
If you analyze those air samples in daycare centers or in hospitals, you have this information, you have catalog almost of all the viruses that are circulating in the daycare center. What's next? What can you do with that information?
Indeed, it is very important to ask this question because we would like to connect this to the clinical surveillance as much as possible. Clinical surveillance or surveillance directly from patients is very valuable for the patients and also for the public. And we want to know which viruses or which variants are actually resulting in more severe cases or in cases that we are scared of. And for this, we need to connect our data to clinical surveillance as much as possible. And if we can, we would like to do surveillance of many viruses at once from wastewater. And this will give us a better view of viruses circulating. So that is our overarching aim, I would say.
indeed you take air samples and wastewater samples. They are complementary, or they are two ways to monitor viral spreading?
So I don't like to call it spreading because we do not know what we detect is actually infectious. That is important to say, but I can say that they are complementary. And in our last study, actually, we show that wastewater captures a very broad diversity of viruses, thousands of viruses. But if it comes to indoor air is more human specific, or can be more human specific if we test correct locations. For example, in emergency department of UZ Leuven, we were able to identify more respiratory viruses compared to wastewater. So indoor air can be more sensitive or more changeable because you can put indoor air sampler to different locations. You can target different locations and you can find more respiratory viruses, which are causing bigger problems than many other animal or skin-related viruses that we may find in wastewater. So I would say that they are complementary, and wastewater is a broad surveillance tool. Indoor air can actually target different populations. And for example, during the COVID pandemic, we had to close different locations during different periods of the pandemic, but there was very little evidence about circulation. So indoor air actually could give us this information. What is the circulation of viruses in different locations?
The analysis of indoor air is more detailed. There are only 20 children and some parents come in those rooms. Wastewater, can you put a number on that? How many people are you monitoring at that time if you're analyzing wastewater?
So our samples are 24-hour composite samples that we collect with our collaborators. And this sample collects wastewater from more than 100,000 people all around Leuven. So depending on how many people are using their water and they're going to toilets and so on, it highly depends on that, of course. But I would say around 100,000 people are that we can do surveillance for. For indoor air, it's highly variable. It can be 20. Or you can put the indoor air sampler to an HVAC system and do a surveillance for a whole very big building of 1,000 people. So it's not defined that it has to be 20 people necessarily.
Using this method, you could detect the parvovirus B19. You could detect it almost a year before public health alerts came. Why is that an important illustration of the value of this method, of this viral metagenomics?
For example, for parvovirus B19, we know that it causes only mild infections in children and some adults, but it can cause some complications in pregnant women. And this is important because this is not a virus that is tested continuously for everybody who's getting sick. As it can show as a respiratory symptom. But it's important to know because these complications in pregnant women can actually be returned if we can detect it on time. And in our case, alerts were probably shared after a year because pregnant women get sick and then there's quite a bit of time to see the results. So there can be complications with the babies and so on. So in the end, after a year in Western Europe, there were different reports and also news articles showing that there were issues about this virus. But in our data set, it showed that we detect parvovirus B19 starting from 2023. And this was important because nobody would have tested this with very targeted methods such as PCR. But metagenomics allows us to find these viruses that we think they're not that important, but can still cause complications in certain populations.
In this example, you are looking back to the source of spreading of a virus. Can you do it the other way around? If you find some virus in wastewater or in air samples, that you can start like a vaccination campaign or something to prevent outbreaks of happening, maybe so you don't predict them anymore because they don't happen because you warned people early enough.
Yes, actually, there's a good example of that from our collaborators. This was targeted because it was done for measles. So our collaborators, Mark van Ranst and Elke Wollens, they were looking at Brussels wastewater and Leuven wastewater to see measles, if there was measles present. And actually, measles is one of those diseases that you can run vaccination campaigns for. Or Mpox, for example, that you can also detect in wastewater. Obviously, these are very high importance pathogens, so people try to test for these viruses immediately, but there will be some viruses, indeed. This might be useful.
Can you also use this technique to predict which variant will be big in winter and so adapt the vaccination? And if I'm looking very far forward in the future, maybe eliminating the seasonal flu in that way?
I would say it's very hard to eliminate seasonal flu, probably, but it is a very good point that we will be able to see which kind of influenza, which kind of flu is circulating the most before the season begins. But this does not necessarily mean that those variants will be dominant in winter. So that's the tricky part about virus epidemiology. We cannot say for sure, but different subtypes showing that we do not expect, such as H5N1, it was a very hot topic. If we found it in wastewater or indoor air, it would mean a lot of things. That we should do better surveillance on different locations maybe or different populations, because we will probably detect it in wastewater before it goes into the hospital.
Which challenges, scientific challenges, still need to be overtaken to make this, to realize this, to make like an early warning system that can totally prevent new pandemics, epidemics?
So of course there are scientific and practical challenges that has to be addressed. But for scientific, I would say the main issue is comparability between different sites for indoor air and wastewater, because we know what's happening in Leuven. So I do know if I see a new variant, what will that mean? And we are using different mathematical models or different correction methods to be able to say which virus is now on the rise. There are potentially more cases, but this is very hard. So actually connecting to real cases in the hospital, that is the most important thing. That's the thing that we are trying to do right now. And also practically, we need more sites and comparability, and we have to know what is going to happen in the clinics if we see these viruses in certain locations.
Interest in virology was huge during the COVID-19 pandemic. Everything about in the news, everything in science news was about virology that time. But now it seems the interest is fading. Do you feel that?
Well, I do feel that, and that is kind of normal because there's no urgency anymore, of course, about viruses. It does not affect our lives immediately the way we feel it, like in the COVID. But it is important to mention that these systems that we are trying to build are kind of smoke detectors, because we want to have a system that is available to detect viruses before they cause big problems or before certain variants are actually establishing themselves. So it is important to keep our attention, especially for researchers and for people who are in charge of these decisions, that these systems should be funded well. Like every research question, I'm sure everybody would like to say that, but this is very real, and we do not have less chances of getting another outbreak after COVID-19.
COVID-19 pandemic started more than 7 years ago now. Are we better prepared for a next pandemic now than 7 years ago?
In many ways, I would say yes. To start with different testing approaches and different testing. I'm sure the testing capacity of many hospitals all around the world increased so much, which is a very good thing, because to be able to answer these kind of outbreaks, we have to test these viruses. And of course, the technology and the funding during the COVID-19 opened so many doors for researchers to fight these kind of pathogens. But of course, it is not done. And for pathogens, it's hard to say it will be ever done.
I heard a lot of interesting things, a lot of fascinating science, but I also want to ask some personal questions about you as a researcher. So you were born in Turkey and you moved to Belgium for your PhD. How was that like?
It was great. So in 2021, I came here for an Erasmus exchange. I did 5-month Erasmus exchange, which I was working as a trainee in the lab, that I learned so many microbiology techniques, and I always wanted to do research. So Belgium was a great place for me to do research, which as far as I could see at my Erasmus time. Then I came here and I'm very happy to be in Belgium. I have to say, the research environment and the high quality research. Yeah. It's very nice to be here.
How you like Leuven as a city to live in?
I do love Leuven because it's a small but vibrant city. I can find everything that I want, but also I do have my friends. I have my partner who is a Belgian. So I do like living in Leuven and being part of Leuven and KU Leuven.
You trained as a medical doctor, so normally you would end up in a hospital helping patients, but you ended up in research. Why did you make that switch, and what's maybe the value of having that knowledge as a doctor to use now in the research?
So this change in career actually comes from, I guess, my childhood, because I was always inspired by nature, and I remember that I was looking at encyclopedia images, and I was collecting seashells from the beaches all around Turkey and trying to identify them, looking at encyclopedias. And after that, my interest grew into more being a doctor and medicine, but I never lost my appetite for research, and I always wanted to know more, investigate, make sense of things, even if it didn't make sense. So now I wanted to do research full-time instead of being a doctor and being able to do research half-time, because being research full-time actually has its own advantages. Like, I'm able to focus on what I want to think and what I want to write. And I see these two different domains as very complementary, because medicine helps people immediately to your close circle, to people immediately that you see, and research actually helps the society and whole world, if we think about it that way. And that I do like thinking about them together. They're definitely complementary in my mind.
That's a great idea. And then my final question, what do you hope to realize in the next years, maybe in the next decennia in your career?
I do like being a researcher. So I want to continue doing research, that's for sure. About the topics, we do have some ideas, which some of them are connected to these kind of environmental surveillance approaches, viral metagenomics. And I also think about going back to medicine to do a clinical microbiology specialty, which I do like the idea, because I would like to have that clinician look during my research as well. I think there's so much that we can achieve if we can collaborate as good as possible. And I would like to be that person in between who can do viral metagenomics, but also understands patient sides and usefulness of these kind of methods.
Okay, I wish you a lot of luck in achieving
Thank
that.
You so much.
And realizing that. And thank you for this very interesting podcast.
Thank you so much.
En jij, beste luisteraar, bedankt voor het luisteren. In deze aflevering maakte je kennis met een van de 5 laureaten voor de Eos Pipet 2026. Ben je benieuwd naar de andere laureaten? Beluister dan ook hun podcastgesprek of surf naar www.eoswetenschap.eu/eospipet2026. Daar kan je trouwens ook stemmen voor de publieksfavoriet. Dag!